I want to welcome you to this session. My name is Rose Banzaf. I work on several volunteer organizations in environmental education across the state. But my major role right now is on the Project Learning Tree Advisory Board for Colorado. I would like you to know that if you have any emergency tech help that you need throughout this session today, I will be putting Nicole's phone number up on the chat box for you to be able to call her in for help. I want to thank you for joining us, and I will be your host for this session. And we would like to thank all of our Advancing Environmental Education Conference sponsors who have continued with their support through the challenges and changes we've occurred over the last few weeks in our community. And we certainly appreciate each and every one of them. To make sure things run smoothly, I have a few reminders for everyone related to using Zoom. Please ensure that you are muted unless we go to questions or breakout rooms. Please add your name to the group chat so we can see who joined us today. If you run into technical problems, please send a private chat to me. Again, my name is Rose Banzaf. So send me a private chat on the chat button below. You can see where it is right down here at the bottom. Or by calling Nicole, and I'll post her number on the chat. Feel free to use the chat function as well to send questions to our presenter. And please be patient with us, as this is fairly new to many of us, including myself. And on that note, I'm pleased to introduce to you our presenter for this session, Melanie Armstrong from Western Colorado University is going to be giving us a presentation on the nature of microbes, and fits very well into where we are now in the midst of this pandemic. So Melanie, it's all yours. Take it away.
All right. Thank you, Rose, for that introduction. I'm just sending out several links in the chat box right now so that folks have reference for those later on. They're in the slides, but I know you won't be able to click on them. So I'll try to keep those coming your way.
And just a note, since those might be links that you might want to access later, I found a function in the chat box at the very bottom where those three little dots are, where you would type a message. If you will click on those, you can save this chat at the end, and it'll be saved in a text format to where you'll be able to access those links at a later time. Sorry, Melanie, I just wanted to make sure people were aware of that.
Cool, yeah, and I'm happy to share slides and things like that too, if it's of interest to anyone. So I'm going to try to share my slides through my screen right now. Let me see, first of all, if I can share my screen.
There you go.
And then, how's that look?
Looks great.
All right. I found that we've been teaching strictly online here at Western for a while now, and I found the strangest thing is the sort of loud presenter voice in my house. So please bear with me while I project at the screen, and feel free to chime in at any point if I can clarify or slow down or any of those sorts of things. So my presentation today, small and simple ways to integrate germs into environmental education, has been so much fun for me to put together. This is sort of a passion project of mine. I want to start just by giving you a little background to what brings me to this conversation today. I think there's a lot of bad information swirling around out there. It's important to understand people's credentials and where they come from, particularly when talking about microbiology and germs and things that may be of risk to us. So I've been an environmental educator since 2001. I worked primarily with the National Park Service, so that's my background in EE. And during a lot of the time while I was working for the Park Service, I was pursuing my graduate degree in American Studies. So I don't have a degree in microbiology. I think that's really important to note here. My academic background is in the studies of culture and human nature interactions. So I've taught in geography and history, and now I teach in the Master in Environmental Management program at Western Colorado University. But I have this interest in sort of scary things in nature and how people respond to things that scare us, whether that's forest fire or wolves, or in this case, microbes. And I'm interested particularly in how we build political systems in order to manage and control those scary things, which certainly seems apropos for the current moment. So that led to my research on microbes, and I've been telling folks recently that, you know, sitting at home, staring at my computer, reading about pandemic disease is pretty much what I did from about 2005 to 2010 when I was working on my dissertation. So it all feels very familiar to me. But that led to the publication of my book called Germ Wars, The Politics of Microbes in America's Landscape of Fear. It's academic, it's theoretical, it's not a fun read, but that sort of is where I come at this conversation, is looking at these studies of bioterrorism and pandemic disease. I also chair the Center for the History of Microbiology, the American Society for Microbiology. And every year I attend those annual meetings, the ASM meetings. This year I won't, as they were recently canceled. But when I attend these meetings where microbiologists share all of their information and knowledge about microbes, I find myself sitting with my computer open, Googling terms about, you know, cell mutation and division, and trying to understand that basic ecology that I learned many years ago as a high school student and undergraduate student. So I'm no expert in the biological processes, much more interested in the human connections to microbes and how microbes really influence us and shape our day-to-day lives. So that's what I bring to the conversation. I just wanted to be fully open and transparent about that. And I want to start then with a question today, or a comment really. I see this statement all the time in my work in environmental studies, or something like unto it. Humans are doing more to change the planet than any single species ever has. And I hear this, of course, in terms of climate change, in terms of the Anthropocene, however you wish to describe it. Statement, environmentalist though I am, I often want to throw my hands up and shake my hands and say, wrong, wrong. It's microbes that change the planet more than any other organism. Microbes gave us oxygen, which made this planet inhabitable by multicellular life forms. Microbes, they dominated Earth through most of its geologic history, and arguably they still do. And as microbes evolved, they found diverse ways of obtaining energy. Some oxidize inorganic substrates such as sulfur or iron. They really have generated these diverse metabolic processes that have evolved, and it was cyanobacteria in the ocean that produced oxygen. And due to other shifts in the Earth's surface, over about 200 million years, the amount of oxygen in the planet increased by 10,000-fold. It's a pretty radical change. Thus, you might argue that the most dramatic event on Earth occurred 2.4 billion years ago, when cyanobacteria transformed the Earth to its present oxygen-rich status and triggered the evolution of multicellularity, which is what has brought us all to where we are now. So why study microbes? This is perhaps the current moment, one of those rare but scarcely unheard of moments in time when microbes take center stage in our lives, and their importance and their power and their consequences seem evident. But I want to make a case today for integrating microbes more wholly into environmental education, along with some ideas for how to do it. So my first argument is that to ignore microbes is to ignore most of the Earth's environment. If our focus as educators is the environment, we need to be focusing on microbes, because life on Earth is and always has been predominantly microbial. Most of the species and biomass on the planet, along with the sheer abundance of life on Earth, is microbial. They are dominant in our evolutionary history. And the elemental cycles that we teach students about, carbon cycles, nitrogen cycles, are fundamentally controlled by microbial metabolism. Bacteria and archaea store about 75% of the carbon stored in plants is stored in bacteria and archaea contained within those plants. And about 10 times more nitrogen and phosphorus are stored by microbes than by plants. So when we're thinking about our climate, microbes play a huge role in regulating that climate. And every plant and every animal, including us, harbors microbes, which are critical to our and their survival. My second argument is that teaching about microbes has high potential to transform scientific understanding. People today have to learn how to make informed decisions about scientific issues that are often uncertain, that are subject to interpretation, or they clash with public values and worldviews. And we see how important this is with COVID-19 and the events that we're watching in the news today. People's understanding of science is more critical now than it ever has been. And my own biases here, I recognize how environmental educators are on the front lines of these conversations about science. Scientific interest in microbes has grown steadily since the discovery of microbes, but the general public understanding has not followed suit. So here's a chart showing the number of books published on animal ecology, microbial ecology, and plant ecology as a percentage of books. And you can see that microbial ecology interest is growing, but we're not following suit with our sort of broad understanding, or as the scientific interest in microbes is increasing, our public knowledge is not following suit. And this is where I really see environmental educators as leaders in these conversations, because we have such an adept way of reaching learners of all ages. We're one of the few organizations that really accesses adult learners. And environmental education is a leader in citizen science work, which I think is hugely important in the microbiology world as well. My third case for why studying microbes is that ideas about nature do influence our environmental policies. And this, of course, comes from some of my work that I've done looking at how the relationship between humans and nature influences the environmental policies that we adopt. And if microbes are missing from our conceptualization of nature, it's going to be absent from those relationships. And so again, as we look at our policies and the management practices at play now, how much would a broader recognition of microbes as part of nature shaped the political policies that are now currently shaping our lives? And I think our environmental policies, there's danger in having them bound to this kind of precarious, or to have them precariously bound to this incomplete worldview without microbes. But I think, because microbes aren't visible, maybe they're more abstract. We know they play a key role in ecosystem services, such as carbon sequestration and decomposition and water purification. And so maybe there's also potential here that by broadening our understanding of microbial ecology, we can help the public understand the uncertainties that are inherent in climatic and ecological debates. Microbes, because they're unfamiliar, because they're unseen, as we can begin to understand them better, perhaps our environmental policies will be better suited to address the challenges of a dynamic and changing climate. Finally, I'm going to argue that microbes overcome some of our access issues that we are so concerned about within the field of environmental education. Microbes perhaps can bring justice to EE and overcome some of these access issues. For years, we have focused on showing that nature is in our own backyards. Well, nature is in our own bodies, and we are continually engaging with ecosystem processes happening in our bodies, in our homes, in our local environments all day long. So this takes the kind of nature is everywhere argument to every human being on this planet, every pet on this planet, every plant and every windowsill. And it appeals perhaps to the innate appetite of children to engage with nature by emphasizing how nature and its diverse communities are found on their skin, in their mouths, and in their guts. So beyond that, my case for why study microbes, I think there's value in recognizing what microbiology can also bring to our study of the other ecosystems. Microbiology today is really done with an ecosystems perspective. So the human body and its associated microbial communities are studied using an ecological approach. Thus, scientists are really interested in seeing how the organisms that live within our bodies are interacting with each other in symbiotic ways, in transmitting beyond our bodies to other bodies. An ecosystem function, it's important to remember that it happens at many different levels. And I'll just reference one study that I find really exciting, sort of dealing with this ecosystem approach. We've had in microbiology, scientists for years have been studying the human microbiome, this idea that we go about our environment gathering up different microbes that we then integrate into our body, and every human being has a unique microbial characteristic within our bodies. This study looks at bees and argues that that principle of the microbiome can be scaled up, that we actually share microbiomes as social beings as well. So bees, who are very social, very close to each other, actually co-create a microbiome that is socially created. Those move around the world, gather microbes, bring them back together and share them with each other. And then that evolves over time as the bees pass it on from generation to generation. Ecosystem approaches are certainly familiar to us in the environmental education world. However, they definitely have a macroscopic bias. About the only time where you'll see microbes addressed in ecology textbooks are in discussions, like I said, of the nitrogen cycle, perhaps some examples of symbiosis or life in extreme environments. But this conversation can be made so much richer, even if it does make it a little more complex and uncertain in certain ways. But by recognizing the many ways that microbes operate in ecosystems can certainly enhance our social understanding of scientific information. So if we want children or adults to understand the complexities and intricacies of ecosystems, what better way than to show how our own body, our own schools and parks, homes are microbial systems. And we want to engage students by emphasizing the self, right, as an entry point into environmental education. What better way than to show children that their bodies are in fact entire ecosystems themselves. My final case for the value of this conversation really is that microbes inspire awe. I think there's something so tremendous about recognizing and seeing microbial environments, and we'll see some examples of this in a few minutes. But I think beyond that, there's this sort of mystery and unknown that we can access today. We have so many ready technologies available where we can actually see microbes in ways that could only have been imagined a generation ago. The creator of the first microscope is a man named Antoine van Leeuwenhoek. And it's a wonderful story. If you have an opportunity to read it, I'll tell it in brief right now. But he was just a curious person, a tinkerer, if you will, a hobbyist. And he began to play with lenses. He was fascinated with lenses. And that led to these sketches and this idea of putting multiple lenses together to create a microscope. And so he builds this homemade microscope. And the first thing that he does is he scrapes the plaque off of his teeth and puts it on a microscope slide and looks at it through his homemade microscope and sees things moving on this slide. And I love to imagine what he thought at that moment, right? Nobody knew that microbes existed, and he scraped his teeth, right? This is a few hundred years ago, right? His teeth were not clean, I guarantee. He scrapes his teeth rubs it on a slide and sees things crawling around on that slide. And you can see his excitement in his account because he starts running around and scraping other people's teeth and gathering microbes from every place that he could possibly think to find them, and then showing the neighbors, and it becomes this wonderful gathering of people looking at microorganisms moving on a slide from substances he had been pulling from the environment. And as it happens, he happened to run into, in one of those sort of stories of how science happens, he ran into a much more esteemed acquaintance who worked for the Royal Society and was able to make his case and send some of his drawings into the Royal Society for evaluation. And these are some of the first sketches and images that we have of microbial life came from Van Leeuwenhoek's drawings of these animalcules, he called them, that he saw swimming around on the microscope slides. And I think that this is the power that microbes have to build excitement, to instill that sense of awe. And just as one sort of brief example of this, I want to show a clip of a film. It doesn't have sound. Hopefully it's playing for you there. Just as an example of how microbes can inspire awe, this is a tardigrade, so not a microbe per se, around a grain of sand magnified 200 times. I just love that image. I love he lifts up and he waves at you, and I can imagine a child seeing that through a microscope and just being astounded. That video was made by someone, a home microbiologist, just playing around with microbiology. So my first example of really cool things you can do with microbes is one that's really just for fun, but some of you may have heard of dinoflagellates, which are these bioluminescent organisms. Part of their metabolic processes generate light, and so you hear of them primarily in oceans, right? If you're in the Caribbean, New Zealand, other places where you swim in water and you can run your hands through the water and these organisms glow. Did you know, though, that you can actually order them online? You can buy these dinoflagellates. You can have them shipped to your house. You can put them in a jar or a lamp or a bowl and shake them up and have this sort of DIY glow lamp in your house. If you take care of them, they'll stay alive for several months. So what a fun way to engage children in microbiology, by having these organisms that you can talk about their metabolic processes and see them happening right in front of them. And through the amazing world of online shopping, you can literally have them ordered and delivered to your house. So I want to talk just for a moment, while we're on the subject of awe, I want to talk a bit about children and microbes, and I want to hear in a moment from you your thoughts on what we can do to really bring children into this conversation about microbiology. But a few things that the research has shown about children in microbiology. Children actually really struggle to draw microbes, which surprised me given how engaged children are in drawing and the numerous monsters and things like that that we see. But drawing and conceptualizing the different parts of a microbe, recognizing the differences and concepts of scale are actually quite difficult for children. So as a simple activity that we can do as educators is helping children through drawing to understand the microbial world. Children also know that microbes are everywhere. That's a concept that's actually really easy for them to grasp. The image on this slide is from a scientist who literally took her child's hand and cultured the microbes after he had been out playing in the playground. But they have that sense and that knowledge that microbes are everywhere. But the knowledge that they tend to lack is that picture of the interrelationships, understanding the importance of microbes, understanding how they are related to their own health and survival. That seems really, really difficult for children to grasp. So, young children largely consider microbes as like a strictly human problem, rather than seeing microorganisms as part of an ecosystem, or as pathogens for other organisms beyond humans. Which fits with sort of our understanding, students thinking of humans as the center of the ecosystem rather than just one member independent or interdependent with other organisms. But I'd like to hear from you for a moment. We can do it by chat, or if folks want to unmute themselves and speak up. What else do you think is important to consider when we are teaching children about microbes? What's part of this conversation that we need to tune into?
One thing that occurs to me is that kids are afraid of things that are invisible, things that they can't see and understand. And so introducing them to the fact that there's these invisible things, we need to do it pretty cautiously so that we don't include a piece of fear, or maybe we're able to dispel the fear that might come with that.
I like that. See a couple comments in the chat. They need to know why they are important, how they can connect with the topic.
I would add that microbes, we could teach kids how that microbes are part of our digestive system and make us healthy, and eat, and then you could, microbes and yogurt and cheese and like celebrate microbes in this way.
We could make yogurt.
Yes.
Absolutely.
But make it
And
really positive, and like part of us.
And there's so many interesting microorganisms that help us clean our wastewater too.
Yeah.
I like this comment from Anna that microbes are great for inquiry activities, which open new conversations.
I was going to say on the luminescent ones, I've done this, I've gotten those kits for my grandkids. It never occurred to me, even though we have microscopes available in the house, to put a drop of that onto a slide and let them look and see what's happening. Now I have to get another set.
Good excuse.
Lisa, that's a great one, because we have worm boxes and we talk about the decomposers because we have compost. We have a farm at the J along with forest school, and the kids love to go spread the compost. So that's awesome to talk about the soil microbes more.
Right.
So another comment that came through the chat box in passing was around why we call them germs. And just a quick sort of etymology there, the term germ actually comes from the same root as like germinate. So the origin of the term germ has to do with giving life and starting life. So we've assigned this negative connotation to it now, but it actually did not originate that way. So circling back a little bit to this concept of self and centralization for children within the ecosystem, I'm going to do just a quick activity, a fun one, with a quiz. So take notes, follow along here. So you'll select one of the options here, characterizing yourself. Do you prefer to play inside, or do you prefer to look out the window or play outside? So based upon your response to that, go on to question 2 or question 3. Now, based upon your response there, go on or 5 or 8 9. Now keep track of these. If you are on 5 or 9, note your page number. If you're 4 or 8, you have another question coming your way. But it's going to switch screens, so make note of which number you're looking at. So in that chat link, and I'll put it up again, is a link to this activity, which comes from the American Society for Microbiology workbook. So if you want to open up that PDF document, you'll be able to go to the appropriate page at the end and identify which microbe you are most like. So we won't spend too long on that, but go ahead and try to pull it up, see if you can find your light microbe. Let us know in the chat box which one you are.
So I was supposed to go to page 12 and it ended on page 9.
Oh no. Sorry, I'll pull it up, see if I can. Yeah, my link, it does, it's the boxes with a, or the pages with a red box around them. They may have their own numbering system.
They do.
Sorry about that, a little confusing there. We got a lot of streptococcus and staphylococcus in this group, those outside loving organisms.
All
right. In the interest of time, I'm going to push on, but you have the resource there. You can learn all about which microbe most fits your personality. And I want to cover real briefly here then 4 keys to effective microbiology education. My first argument is that we have to work on making the invisible visible. And there are lots of resources that we have for doing this, but making the invisible allows that which is abstract to be seen and smelled, and it results in that kind of multi-sensory training, which is really effective in memory formation and recall. And so agar work is really easy to do. You can actually make your own agar medium, or you can buy agar plates on Amazon or wherever. They cost a couple bucks a piece. And it's really easy to simply, mark them off, swab things, put them on the plate, the medium, and allow them to grow. Really easy to do and make comparisons about different places and the microbes that you find in different locations. And really, it's a great exercise for honing those skills of observation, comparison, deduction, helping students to really understand the different conditions that cultivate or discourage growth, applying that to understanding of environments. There's another great resource. It's in the set of links that I texted earlier to this guide. That's just a practical microbiology for secondary schools, comes out of the UK, but just talks, has a good safety briefing at the beginning, but then a whole range of about 15 different simple experiments that you can do with agar work. When you become really adept, you can practice your agar art. This has become quite a phenomenon in the microbiology world recently, but by cultivating microbes of different colors and different growth environments, you can really get into the artistic, creative side of things. So the American Society for Microbiology has a contest every year for agar art, and these are some of the winners over the last few years. Don't discount microscopes. Microscopes are increasingly affordable. This one's a simple microscope that links up to your computer, even your phone. Costs 23 bucks and can get significant magnification and allow you to really start to see. Here's yeast at a magnification of 400 times, where you're starting to see yeast cells in water and the little buds. That's the way yeast grows and divides. And then, of course, don't forget the simple basics of growing microbes, however you do best. I'm certainly doing this in my refrigerator right now. But certainly, you know, food products, but then also these more structured ways. The Winogradsky column is sort of your traditional study of pond scum, right? Where you put pond scum in, or pond water, pond scum first, and then soil, and then pond water into a column, feed it different things, and watch the different organisms that grow within those columns. So consider environmental activities that help to make those microbes visible as a starting place for any conversation about microbes. The second principle that I would strongly encourage is really working to break away from assigning good and bad values to microbes. As we've come to kind of embrace our microbiome, we've slowly allowed microbes to become seen as a necessary or even desirable. But these categories still perpetuate the idea that microbes are either good or bad. And in reality, they're living organisms that have complex lives. I don't know, you can argue that they're that complex, but they are working for their own survival in ways that all organisms do. And categorizing them in these two categories probably doesn't serve to really have those nuanced ways of thinking about microbes that can lead to broader social and political practices. So we see, you know, evidence abounding that people are starting to embrace the microbiomes. You can buy bacteria online to restore your bacteria to your skin. You buy probiotics. There's the Back to Dirt movement, where you're supposed to expose your kids to dirt. All of these things are really promoting that positivity. And certainly that's a place to start with this conversation. But I challenge you to go a little bit beyond that in order to have to bring the same complexity to microbes that we would bring to any conversation about environments or ecosystems. So as several of you mentioned, a great place to start is with cooking. So this link is also available in that set of links to a sourdough storybook. This is a time when sourdough is all the rage. Everyone's cooking at home and building their sourdough starters. And there are instructions on how to gather your own yeast and develop a starter. And then also experiments that you can do with yeast and sourdough contained within this booklet. So a fun place, you know, thinking about how we start with food. Also draw your attention to some citizen science work around sourdough. I don't think this project is currently collecting samples, but for a while they were asking folks to send their sourdough, and they were doing the sequencing to understand the genetic variations and the types of yeasts that were being collected all around the world. So a great example of citizen science. And we've talked about, you know, making your own cheese and yogurt and sauerkraut and beer, all of these projects that involve microbes. While we're on the topic of citizen science, just a couple other opportunities to get involved. The American Gut Project will take a sample of your microbiome, and they're the largest crowdsourced collection of human gut microbes, and one of the largest citizen science projects in the country. For those who like on the other end, there was this wonderful project about donating your feces for folks who were looking to study the Open Biome Project. I actually took this picture at a bus stop in Boston, inviting people to donate their stool. So anything is possible. So in that realm of looking and promoting positive uses of microbes, I think that idea of looking to utility and helping us to find microbes in our everyday world is useful. So we're going to go for a little activity here. And I want you to select one of these organisms. They're not organisms, objects. Select a license plate, vitamin D, laundry detergent, ketchup, or a peanut. And we're actually going to break you into breakout rooms so that you can spend some time talking in small groups. And so what I want you to do is identify the microbial property that is associated with this object. So find out what microbes have to do with this project. And then, and I want this to be a literal scavenger hunt, go find as many additional items in your house as you can that were made through that same property. So Googling is fine. You'll have to figure out what it is that microbes have to do with license plates. And we'll spend, we'll do this pretty fast in about 7 minutes in each group, and then we'll have a couple groups come back to show and tell. I think because we have a pretty big group today, I'm going to go ahead and ask Rose to divide us into maybe 10 breakout rooms, and go ahead and just pick whichever object catches your fancy of those.
So Melanie, we are at about 5 minutes left, and
I think we go till 3:15, Rose.
Oh, okay. And I, for some reason, do not have breakout group options.
I do.
Katie, do you do it? Can you do it?
I do. So you should get a message up on your screen and go ahead and click join the room, and you'll be able to move to your breakout room.
Thanks, Katie.
Absolutely.
Hello, is anyone else on?
Hello.
Hi, I'm Lisa from CAEE.
Hi, Lisa. I'm Gabby. Nyampatika. Oh, there we go.
I know I'm in my bedroom. I guess we can video.
Hello, is my microphone working?
Oh yay! Yeah.
Hi.
So what did everyone choose for their object?
I did the license plate.
I did the license plate too, and I was just about to Google. Have you found anything yet on that one?
I was trying to think of like anything associated with that. I was kind of confused by that.
I think so. I'm thinking like dirt that may have come off the road that kicked up.
Oh, like, okay.
But I don't know. I could be off track here.
It has to do with like if there's a process that the microbes have been in that helps the metal bond together or something, like in the actual process of making the license plate. I was doing the vitamin D.
The vitamin D one?
Yeah.
Yeah, it's hard to see.
I think this is going to take more research than talking.
I Googled like microbes license plate, and I got like license plates with actual microbes on them.
That's what I'm seeing
Is that what
too.
you're seeing too? I'm like, that's what I was going for.
Well,
But
I
if you
guess
wanted a license plate with bacteria on it, I guess they're out there. It's too funny. What did you choose, Gabriella?
I was looking at ketchup, and it was saying how lactic acid fermentation is part of the process, and that is like a oxygen-free process that requires bacteria to do their thing.
Huh.
Yeah.
Cool.
But I'm looking at the license plate now and let's see.
I know, I'm not really finding anything. I have to do a more
Well, it looks like vitamin D is like intestinal health related. So that's interesting. Like, yeah, there's a lot of stuff to read here. I guess for shortcut, I'm gonna have to go with the vitamin D helps with the gut health.
Mm-hmm.
So
Austin, have you found anything else on the license plates?
I found a weird bacteria that I'm trying to look into it to see exactly. It's a bacteria that like apparently like every time it breathes, it essentially produces gold, like very small amounts of gold.
Wow.
Whoa.
It's like metal eating or metal enduring.
Whoa.
It's Capria Vitis Metalli Duran. This I can post. I think I can post it in this chat, right? Does this do that one? I've never used Zoom before. This is the first time I've ever tried it.
I think the chat might go through to everybody, but
Oh yeah, it
that's
definitely
okay.
Yeah. Yeah, I don't know how to. Let's see.
Can you share the screen?
Let's see.
You might be able to.
Can I?
I have the button here for sharing the screen.
I think it's. Is it working? Is it showing my screen?
It's trying to.
Yeah, I think so.
Oh yeah, it worked.
This metal right here.
Cupriolitis.
I'm trying to find more on it, but I'm like, I wanted to see specifically what metal and stuff like that, so I wasn't sure. Yeah, I can. Cool. There we go.
Bye.
Oh, I guess we're going back to the main session.
Bye.
yeah,
Not yet, but in a few.
That's true. We have 43 seconds to go.
I was gonna say we did more researching than talking there.
I know, it's hard to find.
Yeah, we didn't have much time.
Well, hopefully some other people made some progress.
Yeah, I guess I would look for yogurt, and I'm trying to think what else I might like do on my scavenger hunt.
Oh yeah, that's true. We were supposed to, I didn't even pass step 1 on the license plate.
Yeah.
Oh,
All right, I'll see you guys in the main session.
okay, nice to chat with you.
All right, bye.
Connection to share.
Yeah, I kind of found something neat.
All right, what you got?
It was this bacteria that apparently it uses another bacteria in the formation of gold nuggets by precipitating metallic gold from a solution of gold 3 chloride. So it was actually really, it said it was really good in heavy metal situations, like metal toxicity situations. So we chose the license plate, so it's kind of related, but.
I like it. Anyone else find something for the license plate?
Yeah, it's I found something in the reflective coating. It's microbes in the reflective coating on the license plates.
Yep. And on surfaces of roads and places they want to shine as a microbial enzyme.
Right,
you
I
find
couldn't
anything
find
in your
anything in
house?
my house with the same coating.
Find another sign or something.
I found something for the license plates that suggested that microbes might take up metallic elements or take in metallic elements, which I thought was really interesting. I didn't get to read too far on it.
Nice. This PDF here that's showing on your screen, again, one of the links, and Nicole's sending a corrected link for the sourdough as well, takes you to this microbial discovery activity box. And in that document, you can find a list of all of these connections. It'll list the different microbes, and then there's a game at the end that's like which of these is not like the others, and then we'll talk about sort of shared properties between all of those organisms. So another resource to check out and explore. And the last link that I shared with everyone was from the American Society for Microbiology and K through 12 resources for educators, where a couple of these have come from. So just have a few minutes left, so I'm going to keep pressing on a little bit here and you to really think about viruses and this idea of like breaking away from good and bad values. I want to challenge you to rethink the role of the virus. The argument is made by microbiologists and many others that viruses, rather than just existing to harm species, that they've actually co-evolved with species in a way that contributes to our lives. Viruses have a particularly bad rap right now, but they're one of the primary sources of genetic variation, and therefore they are the creators of life. How does it change when we think about viruses as makers of species? So I have 3 cases to illustrate this. The first is around the gene. 80% of the human genome consists of retroviral DNA sequences that have inserted themselves into our human germline, where some of the functions of viruses have been adopted to serve essential functions for our survival and development. 8% is not a small amount when we think about the percentage of our DNA that is directly related to the role of viruses in our lives. In one study, Shami, a microbiologist out of UC Davis, showed that viral proteins are responsible for fusing cells to the placental wall, which mediate the exchange of nutrients between mothers and fetuses. So those same viral proteins are also shown to be responsible for the differences in muscle mass between male and female mice, which is typical of any species that reproduces with a placenta. Thus, our human reproduction relies upon viruses and the traits that viruses have brought to our DNA. A case around cancer. Colorectal cancer is the second leading cause of cancer-related death in the United States and a primary cause of morbidity and mortality throughout the world. And Dr. Jeffrey Hannigan's lab has been studying the virus community of virome contained in stool samples of people with both healthy and cancerous colons. And scientists did not discover any viruses that would be the cause of cancer, but they learned some interesting things about the human virome. While the diversity of viruses remain the same in subjects without cancer, cancer-associated virome contain mostly bacteriophages, or viruses infecting the bacteria of the microbiome. So researchers propose that these phages are modulating the bacterial community, impacting the progression of the cancer. So they suggest that their findings also just suggest an opportunity to diagnose cancer by analyzing microbes. So maybe you could actually just take a stool sample rather than having a colonoscopy, which could be a great benefit to humans as a whole. And a third example is around climate change. The work of Marilyn Rusnick is studying how viruses are enabling plants to adapt to a changing climate. So there are particular viral properties when the viruses are interacting with tobacco plants that enable them to survive hotter and drier conditions. And so you might argue that viruses could be the key to our ability to survive on this planet as the climate warms and changes. A third principle to apply when teaching about microbes is to continually apply these principles of ecology. So consider mutation and evolution. Think about how human species interactions lead to the transference of disease. The microbiome, we tend to favor the bacteria within that, but the bacteria are also infected with viruses and viruses with phages, and there's this whole microbial community. So when we're looking at interactions of ecosystems within microbial communities, recognizing them as also a fierce battleground and competition for resources, we can explore those same principles of ecology that we apply to macro ecosystems to micro ecosystems. And in the interest of time, I'm going to forego another activity here, just thinking about how diseases spread and contact. But basically a simple activity where you are using dots and children moving around in space to understand transmission, and then you can use different colored dots to provide kids with immunity in order to not become carriers. So a fun activity to think about how the disease is transmitted and spread between humans. And the fourth key as we're wrapping up here is to really think about how this connects to conservation. And I know from our keynote speaker this morning, I wanted to go in and scrub conservation from the slide, but I couldn't think of another appropriate word to use. We've been talking a lot about the emergence of COVID-19 and recognizing the coronavirus that causes COVID-19, and recognizing the role of deforestation and increasing conflict between humans and species that comes from habitat loss, and thinking about that as an important conversation to have. When I worked at Yellowstone, I used to talk about the many values of the microbes found in the hot springs in Yellowstone, and the fortunate circumstance that they've been protected, where a lot of other hot springs in the world have not been protected, and therefore we've lost opportunities to study and learn from those microbes. The polymerase chain reaction, the key to studying DNA, came from an enzyme produced by Thermus aquaticus found in the hot springs in Yellowstone. And I like to put out little contradictions as well. In 1980, we celebrated the demise of smallpox, the eradication of a microbe from this planet. It was something that we had spent billions of dollars on doing. How does that contradict with what we currently see in decline of biodiversity and loss of species? How is it that the same species that can actively seek to eradicate a microbe is also actively seeking to protect macro species instead? So there are a lot of contradictions, and I encourage you not to shy away from them and to think about those in educating about microbes. So a quick conclusion here. I think that we continue to manage germs as a problem to be solved primarily through eradication of species, and I think there's a lot more complexity there. There's a lot more that we could recognize, not just as sort of a social condition that goes along with this, but also what it means to live with our microbiome, to live with microbes, and to recognize how vital they are to our existence as species. The fact that we have life the way that we have it on this planet and as individuals is directly attributable not just to microbes, but to viruses, which is a pretty provocative thought in this day and age. And I'll wrap up with just a couple other things. I couldn't find a great place to stick them into the presentation, but I think they're so cool that I wanted to share them. If you are a formal educator, I encourage you to look at the CPhages project. This is Science Education Alliance Phage Hunters Advancing Genomics and Evolutionary Science. You can apply to partner with their project, and it's a year-long project, but they will take you through the process of actually digging in the soil and finding new viruses that they then help do the genetic sequencing on, and then students have the opportunity to actually name, have a virus named after them, or to pick the name for a virus. So this idea of discovering species and giving name and new knowledge to the world is directly applicable through the CPhages project. I've been a huge fan of this project for a long time. I also have a colleague who has a great project that you may have heard of called Self-Made. She's actually making cheeses from microbes from human bodies. So sampling someone's armpit, toe, hand, nose. She's worked with celebrities, so you can get cheese that's made with Michael Pollan's biome. She's doing it as art to display, but secretly I've always wanted to do this in my home, so we'll see how long shelter in place lasts. We may see Dr. Armstrong making some cheeses based upon microbes in my own home. And of course, just citizen science work. SciStarter is a great place, but there are other resources to check out, opportunities to be involved in microbial citizen science, whether it's sending in microbe samples. Here's one where you're looking for microbial communities in rocks. Snow data, collecting samples from snow to look at microbes would be great for here in Colorado. So I'm sorry I get so excited. I talk so long, I didn't leave a lot of time for discussion. I'm easy to find. My email address is marmstrong@western.edu, and I'm always happy and excited to talk about microbes. And for those who are interested, I'm happy to stay on the chat and talk for a few minutes beyond our time too. So thank you very much.
Thank you.
Thanks, guys.
Thank you, Melanie, for offering to adapt your conference session to this virtual presentation and sharing it with us. We hope